Okay, so worksheet number 7. So in this worksheet, we're covering material that's found in chapter 9, which is our last chapter in our protein function unit. And in our protein function unit, we have looked at, of course, things like enzymes and enzymatic catalysis in chapters 6 and 7. And then chapter 8, of course, we looked at ligand binding and cooperativity and allostery, all very important aspects of protein function. And in chapter 9, we're thinking about here another very important aspect of protein function, and that is membrane transport. Okay, when we look at the objectives for our worksheet that we're looking at, you'll see that there are 3 primary ones, and I'll just read them. So the first 2 say use kinetic parameters to compare transporters and predict the effects of mutation on the activity of a transporter. And the activity of a transporter is measured as Km and Kcat. And you think to yourself, wow, that sounds a lot like what we've done before in chapter 7. And that's right, you can't escape kinetics, Km and Kcat. And so we're going to look at some of those things. We're going to, of course, apply those things to transporters. And quite honestly, I think that this gives a really nice application for the use of kinetic parameters. And so we'll look at that. But that'll actually be the second thing that we do. The first thing that we're going to go ahead and do is look at this third one. And the third objective says distinguish between the main classification of transporters. Okay, and we're going to do that. And what you're looking at here would be essentially the structure of a GLUT transporter. Okay, and so when we look at this, really, we're going to be thinking more along the lines of what you see on your left, and thinking about how we can use that structural information to determine what class of transporter are we at. Okay, so what I want you to do right now, of course, is pause it and go through those questions, and then as you go through those questions, after you go through those questions, go ahead and unpause it. Come back and listen to the explanation for this. Okay? Right. All right, so I trust that you've paused it, that you've gone through those questions, and that you've come to a conclusion on those things. Okay, and so the first one, right, would be this: predict what broad classification, primary, active transporter, carrier, or channel this transporter is part of, given this figure, and state a reason why you chose that classification. Okay, so hopefully you come down to a reasonable explanation of this and what classification this is. And I'm going to go ahead and tell you what classification is, and this is a carrier. Okay, now the question is, well, how would I know that given this information? And some of you say, well, I knew that this was a carrier because you told us in the Echo 360 recordings that it was a carrier, that GLUT transporters and that family of transporters are carriers, and they carry out passive transport. Okay, and so that's fair. You cheated. Okay, well, you didn't really cheat, but you get the idea. Okay, you had that information given to you, so you took the easiest answer to that. However, you can look at this. Okay, and I want to exemplify how you can actually take structural information and use that structural information to come to a proper conclusion on what classification of transporter we have, whether it be carrier, primary active, or a channel. Okay, so let me go ahead and go to our next slide here and make sure that I've got enough room on here to write. Okay, there we go, something along those lines. Okay, and when we think about carriers, one of the things to really consider is that carriers will undergo that, what I have called that transportation cycle, okay, where they'll have these 4 different steps that they have to carry out to move something across the membrane, okay, and this would be at a minimum, they have to have those, okay. And so when you think about a carrier, let me go ahead and draw out for us this just generally, okay, and we'll draw, oops, I put it in the wrong orientation, there we go. And we'll just say that that's our carrier to start off with, and we will just call this the inside of the cell, okay, and this would be the outside of the cell, okay, and that's the general shape, if you will, of the carrier. And what we know is that the very first step in this whole process is that the substrate is going to have to bind and be recognized by that particular transporter. Okay, now when that recognition occurs, okay, then there will be a conformational change in the transporter that will open it up to the other side of the membrane. Okay, and so we'll have here our substrate, and now it's opened up to the other side of the membrane, and this would be called the translocation step. So the first step would be recognition, the second step would be translocation. Okay, and then the next step, of course, would be a release step. Okay, nothing too interesting happens here on a conformational level with the protein. However, it's very important that we recognize that now our substrate has been released onto the other side of the membrane, of course. And then finally, the last step would be a conformational change back to the original conformation, what we would call the recovery step. Okay, and now, of course, the cycle is complete, and we can go back to the recognition step, and the whole process can occur again. And that's the process that a carrier would undergo. Okay, and notice that it's important from our discussion that the carrier is not open to both sides of the membrane at the same time. Okay, it doesn't form what we would call a channel, right? So if you look at channels are going to look like this. We'll go ahead and put the membrane here, the in and the outside of the membrane. Okay, and a channel really truly is just that. It's a channel. It's something that is essentially a hole or a pore within the membrane. Okay, that's open to both sides of the membrane at the same time, and it doesn't have to go through this conformational change and this recognition, translocation, release, and then recovery step. None of those exist. Rather, the substrate, whatever it is, can go straight through the membrane, straight through that channel. Okay, now that's not to say that channels can't undergo some form of conformational change. They do. There's gated channels, and they can open and they can close. Okay, but that's not a transportation cycle, if you will. Okay. That we see in the carrier. Okay, so when we come back to our structural information here, right? Let's just say that you don't even know this is GLUT. You don't the GLUT transporter. You don't know anything. Could you look at this and say, yeah, I can make a determination on whether this is a channel or a carrier? And you can from a structural standpoint, because if you think about it this way, the structure of this GLUT transporter is essentially like this, right? Right, and this would be the inside of the cell. And this would be the outside of the cell, if you will. Okay, and you can see that there's this little portion of it that is closed off. Certainly, the GLUT or the glucose molecule, which is actually shown over here on your right, can actually get into a portion of the membrane, but it can't get all the way through. There's going to have to be some opening of this opaque area. Okay, that will allow it to go all the way through. Okay, and maybe I should have, some of you are like, well, I wish you would have told me what the opaque area is, and I probably didn't do as good a job as I could have explaining what you're looking at here. So this transporter, let me just say this transporter is essentially this depiction of a transporter is essentially a transporter that's been sliced in half. Okay, so that gray area that you see there, that's all membrane, or that's all protein structure. Okay, and then this portion that you kind of see in color, that's kind of the beginnings of what might be called not a channel, because I don't want to confuse it with other type of protein, but like a hole, a deep cavity within the protein that allows this substrate to go about partway through the membrane. Okay, and then it has to undergo this conformational change. Okay, so hopefully that helps you out. Now, some of you are sitting there saying, okay, I see what you're saying. I can see why it's a carrier, not a channel, but what I don't understand is why it wouldn't be considered a primary active transporter instead of a carrier. Okay, and that is a very fair question. Okay, if you were given just the information that you see structurally here, could you make a determination on whether this is a carrier or a primary active transporter? And I would say no, you couldn't. Okay, because the only real difference between primary and active transporters and carriers are the use of an exergonic chemical reaction to move something against a concentration gradient, which you can't tell that they'll do from a structural standpoint. Okay, primary active transporters do carry out they have these actually fairly complex transportation cycles, and they're not open to both sides of the membrane, much like carriers. So you couldn't tell just by looking at just this that this is a carrier or a primary active transporter. You'd have to have some other information about the protein. I would have to have other information about the protein. Everyone would. So, okay. Now, the next part of this activity is essentially this, and it says, now draw a velocity or rate graph for the rate of transport of glucose across the membrane versus the concentration of glucose outside the membrane. Does this graph saturate, and if so, why? Okay, and so what we want to do is go ahead and look at this, okay, and come back here and say, okay, well, if we were to draw this graph here, and we're to plot velocity of that versus substrate concentration, what would the shape of this curve be, and would it saturate? And the answer to this is yes, it would saturate, and the shape of the curve would be more than likely hyperbolic. Some people might say, well, could it be sigmoidal? Sure, because transporters can be cooperative or even allosteric, though we didn't really talk about that. Okay, so we'll just leave it as hyperbolic. Okay, but it definitely saturates, okay, meaning it obtains this V max, okay, and the reason is because of the mechanism up here that that it has to undergo or undertake to get something across the membrane, okay. There is going to be a K-cat, meaning there is going to be a rate constant for the rate limiting step of this process, okay, and that will limit the maximal rate that this transporter can move something across the membrane, okay. And because it will limit it, when the membrane transporter obtains saturating levels of substrate, its rate will level off. It will no longer be able to increase the rate of movement across the membrane because, of course, the K-cat is going to limit that. Okay, the rate constant of the rate limiting step is going to limit that. Okay. Okay, so hopefully that helps you out quite a bit. Now, the next one, we'll talk about mutation. Let's put the mutation in our activity number 2. Okay, we'll think about it then. And what we want to do with activity number 2 is we want to actually launch into kinetics, and we want to think about Km, and we want to think about Kcat, and we want to think about all of those types of things, and relate those concepts back to transporters. Okay, and of course we are going to, as we think about Kcat and Km, we're really going over material that we've talked about from chapter 7. It's just something, like I said in the introduction to this, that you can't really escape. Okay, and so we're going to be looking at 2 different transporters. You see them on your worksheet, and that's GLUT1, and I believe your worksheet says GLUT3. Okay, and so we have GLUT1 has a particular Km, okay, for glucose, and then we have another GLUT transporter, GLUT3, that has a particular Km for glucose. And there's a series of questions that comes after that. Understand that both GLUT transporters, GLUT1 and GLUT3, are going to transport glucose across the membrane. Okay, all right. So go ahead and pause it, look at that, and then of course come back and get the feedback on that. Okay, all right. Okay, so hopefully you have figured out the questions and now are ready for some feedback. So let me just start off with this. So I didn't have the worksheet sitting right in front of me. And so on your worksheet, it probably says GLUT2 and GLUT3. I'm sorry about that. I got confused there. And so GLUT3 has this 1 millimolar Km, and GLUT2 has this 15 millimolar Km, okay? And so for the first question is this: which transporter has a higher affinity for glucose, okay? And so hopefully you're thinking to yourself, well, affinity and specificity can be measured either by Km or Kd. In this case, we have Kms because we have things that are essentially acting like enzymes. And so we look at the Km, and hopefully you know that smaller Kms mean more specificity, okay, than larger Kms. And when we think about then smaller Kms, we would think about them as being having a higher affinity for that substrate. Okay, so when you think about, for instance, the difference between GLUT3 and GLUT2, there's a 15 times difference, right, in the Km. So GLUT2 has a Km that's 15 times larger than that of GLUT3. And so in this case, we want to say that GLUT3 has the higher affinity for glucose. Okay, now what do those things mean, right? Well, when we look at these, we want to make sure that we understand that the Km represents the substrate concentration, which we're at 50% of our maximum velocity. Okay, so when you look at GLUT3, for instance, it would take a concentration of glucose of 1 millimolar to reach 50% of its maximal velocity, whereas with GLUT2, it'd take a concentration of 15 millimolar glucose to reach 50% of its maximal velocity. Okay, now that really leads to our next question. At physiological concentrations of glucose, state what percentage of Vmax each transporter will be at within the body, and then plot those two. Okay, so let's do this. Okay, so we want to go ahead and figure out, okay, well, what would our percent Vmax be at 5 millimolar? So just so you have some understanding, when we think about blood glucose concentrations, there's typically range anywhere between 4 to 6 millimolar. With 4, anything below 4, you're thinking about a hypoglycemic state, very dangerous condition. Anything above 6 millimolar concentration of glucose in the blood would be hyperglycemic, and that can be also dangerous. Okay, and so on average, we have a concentration of glucose in our blood about 5 millimolar. Okay, all right, so so when we look at this, okay, let's go ahead and write out our Michaelis-Menten equation before we can determine percent Vmax here. And what we have here is this. That's our Michaelis-Menten equation. Okay, and so what we have, let's look at here, GLUT3. Okay, GLUT3. Let's draw this up here. Okay, its VO would be equal to Vmax. Okay, times the substrate concentration. Substrate concentration is 5. Okay, 5 millimolar. And then we will put here. Let me put it in the wrong place here. Put plus 5 here. Okay, and then our Km is going to be 1. Right, 1 millimolar. Okay, so what that means is that we are at 5/6 of the Vmax. Okay, when we think about this physiological range of glucose concentration, which means that GLUT transporter, GLUT3, is going to be sitting at about 83% of its Vmax. Okay, so 5/6 of its Vmax, or 83% of it. Okay, now let's do that for then, use a slightly different color here. Let's do that for GLUT2. And with GLUT2, we can very easily do the same math. VO is equal to Vmax. And hopefully you see where I'm going with this. 5 millimolar divided by 15 plus 5, which means that we're at 5/20ths of the Vmax. Okay, and that's going to mean that we are sitting at about 25% of the Vmax for that particular transporter. Okay, both of them transporting the same molecule, glucose. Okay, now I think it's really important to take a little bit of a moment here and compare these two. Notice that we are at the exact same concentration of glucose, and look at the difference in terms of the rate that we're obtaining for these two different transporters. One of them is sitting at about 83% of its maximal velocity, okay, and the other one is sitting at only about 25% of its maximal velocity. Okay, so, and that's all based off of course, the efficiency, or really not the efficiency, but rather the specificity of the enzyme for that particular substrate. Okay, now when we look at this, there's a couple things more that we want to do. We want to make some applications here, but we also want to do a mutation. Okay, and so let's go ahead and let's do the mutation first. and then we'll look at the application, okay? The application is super awesome, okay? But let's look at the mutation. Mutations are important to understand because they can cause many genetic diseases, but they're also good ways to try and understand and test ourselves whether we understand what KM means and where it is, what physically causes KM, okay? And so I really should come back up here, okay? What we want to think about with the mutation is this, okay? We have this guy right here, okay? This is circled with circle in your worksheet. It says, okay, we'll mutate that. And we're going to mutate that from an asparagine to, it says a leucine. You could do a valine either way, leucine or valine. One of those, either one of those is going to work for our understanding of this. Okay, but when you look at this, the important thing about this asparagine is that it's forming hydrogen bonds to the substrate, right? So this would be our substrate here. This would be glucose. Okay, and then we have those hydrogen bonds that I've circled, and that's what asparagine provides. Well, if we come down here, asparagine side chain is going to be this. And you all say, yes, side chain structure. Asparagine is polar ASN, and that's what it looks like, okay? And we have, as you hopefully probably notice, in that nitrogen group, we have hydrogen bond donors, and in that oxygen coming off at the end of the side chain, we have a hydrogen bond acceptor. Okay, so they can form hydrogen bonds, right? Compare that to valine or leucine or whatever. Let's just use leucine because that's what is discussed in our, or that's what's used in our worksheet. Leucine is going to be this. Okay. And the important part of this is going to be these methyl groups sitting at the end of this. Okay, so when we look at here and here, we have hydrogen bond donors and acceptors. Well, when we look at leucine, we have methyl groups, and those methyl groups aren't hydrogen bond donors, nor are they acceptors. And therefore, this leucine won't form hydrogen bonds, and we'd expect then Km to go, hopefully you all are thinking, up. Okay, it's going to lose affinity or specificity, so Km will get larger because you lose hydrogen bonds. And remember, specificity, there's a physical reason that Km would be low, okay, or small, and that a protein or an enzyme or a transporter would have a high specificity, and that's due to the non-covalent interactions or the binding energy that it can form with the substrate. Okay, in this case, glucose and hydrogen bonds are important in that. Okay. All right, now, the last thing here then, let's come back up and do this application. Like I said, yeah, we'll come right to here. And the application is this, okay? It says which transporter, it says this, GLUT3 is found expressed in neurons, whereas GLUT2 is found in the pancreas. Why do you think it's important for GLUT3 to be expressed in the neurons rather than GLUT2, okay? And so that's an interesting question. And it's a really great application, I think, here to KM and the importance of KM. So it turns out that you and I are going to consume somewhere between 160 to 200 grams of carbohydrate on a daily basis. Okay, about 85% of those carbohydrate molecules that are going to get consumed by us on a daily basis are going to get used by neuronal tissue, in particular the brain. And so when we think about the major user of carbohydrates within our bodies, it is really neuronal tissue, okay, in particular the brain that uses it, okay. This is why maintenance of glucose concentration in the bloodstream is really important. It needs to remain at a fairly high concentration so that the brain is getting the consistent source of energy it needs to continue to run. Of course, if we run out of energy in the brain, that will kill off neuronal tissue in the brain and we become brain dead and dead. Okay, so that's not a good thing. And so when we think about that, then it's interesting that the neuronal tissue comes with and comes equipped with GLUT transporters that at physiological concentrations of glucose, they're at near their maximal value, meaning that the brain essentially sticks a straw into the bloodstream and starts to suck glucose out of the bloodstream irregardless of what other tissues need. Okay, that GLUT3 transporter has a much higher affinity and specificity for glucose, and therefore the brain is going to absorb significantly more glucose than other tissues, like let's say the pancreas that's using GLUT2, right? GLUT2 is only at about a quarter of its maximal velocity at normal physiological glucose concentrations, okay? We'll talk about the pancreas later on and the important role that it plays in the maintenance of glucose concentrations, okay, in particular in the release of something called glucagon and insulin. But for now, we'll just say that it's not like the brain. It's not going to have the specificity for glucose. It's not going to suck down the glucose for its own energy needs because it doesn't need it, whereas the brain does. Okay? All right. So hopefully you found these things, I think, interesting, helpful to you to understand, of course, certainly membrane transport, but also, as we have been discussing, kinetics and the application of Km and Kcat. So we didn't really talk too much about Kcat, but certainly Km. Okay. And if you have any other questions, please let me know.